So, welcome to my channel Future Academics. Today, I am going to explore the polymerase chain reaction or PCR. This is a powerful recombinant DNA technique which is widely used in both basic research and clinical sciences. In this video, I will walk you through its definition, core principles, methodology, mechanism, applications, and critical limitations. So, be with me and watch till the end. So what is PCR? How you define PCR? As the name indicates, polymerase chain means repetition of DNA synthesis reaction again and again in a cyclic manner using the same reaction mixture. Normally, an enzyme reaction cannot be repeated using the same reaction mixture again and again, but it occurs in PCR normally. Therefore, PCR is defined as DNA synthesis done repeatedly multiple times to amplify a target DNA in large amount. Why and how DNA is amplified will be discussed in detail, but for now you should remember that one of the objectives of this technique is to increase the amount of target DNA so that the analytical techniques such as gel electrophoresis, DNA sequencing, or southern blot analysis can be applied. So why do we need PCR or why you use PCR? There are three reasons. The first one is that it is a super fast and super sensitive technique. There is no match for its sensitivity. It can detect even a single molecules from the sample. Not only that, it can amplify the amount by million, billion or trillion fold depending on the level of expression or level of amplification you choose to do. The second reason is any region of a target DNA can be selected using a pair of primers to amplify. Therefore, it provides flexibility in operation, and this property or quality is especially important for the situation where the quality of sample isolated from tissues is compromised. So it means it increases the chances of success of the reaction. And the last one is the amount of target DNA is amplified so that the classical technique or analytical technique can be used without any problem. So the underlying principles, it is based on the principle of DNA synthesis and nucleic acid hybridization. So DNA synthesis is same as DNA replication. Its requirements are same more or less. And nucleic acid hybridization here refers to the binding of two unrelated DNA. In PCR, it refers specifically the binding of primer with its target DNA. So you should be able to identify when and where DNA synthesis happens and when and where nucleic acid hybridization occurs. So the following are the components or the requirements of PCR. Number one is template DNA, primers, substrates or DNTP, and Taq-DNA polymerase. I will elaborate on each of these components in the next slides. Regarding the requirement of template and primer, couple of points. Template is a double-stranded DNA having two strands, strand 1 and strand 2, and this act as a template. It provides the region to be amplified using a set of primer or pair of primer. So there are 3 points you have to remember about the primer. Primers are single-stranded DNA molecule complementary to their target strand. So primer 1 is complementary to strand 1, primer 2 is complementary to strand number 2. The second point is they initiate DNA synthesis. They prime DNA synthesis by providing 3' OH. This is very important point. And the third one is they also define the region to be amplified. So these 3 points you should remember about the primers. PCR is an enzymatic reaction, therefore the next requirement is substrate. These are deoxynucleotide triphosphate. Structure is shown here. There are 4 types: DATP, DCTP, DGTP, and DTTP. Collectively, they are called as DNTP. The structure is shown here. The difference is at the back here. It could be any of the 4 bases. The function of these is also very important. They provide 3' OH for the extension of DNA synthesis. So the function of DNTP is different than the primer. The last requirement is enzyme, Taq-DNA polymerase enzyme. This is special enzyme, and this is isolated from bacteria, thermophile bacteria called Thermus aquaticus. And the enzyme isolated from this is known as Taq-DNA polymerase. This is heat-resistant enzyme. You will notice that in PCR there is a denaturation step, and no enzyme can survive the heating at 90 or 9 above degree centigrade. Therefore, you need heat-resistant enzyme, and therefore, before the discovery of Taq-DNA polymerase, PCR was not practically feasible. So these are 3 points about Taq-DNA polymerase you should remember. So how to set up the reaction or methodology? Take a tube and add the template. Template I have shown here lambda DNA, which is a double-stranded DNA, and segment has been selected by choosing or designing a pair of primers. So add primer also, and then D and TP here, and then Taq-DNA polymerase. Put all these requirements in the tube, amplify for 20 to 40 cycles depending on your need. Perform gel electrophoresis, stain the gel, and see under UV light and on the UV transilluminator. What you will see the amplified fragment only, which is corresponding to this region. So basically on the gel you will see only amplified fragment, PCR fragment only visible. The template will not be visible. This is simply because the amount is below the detection limit. So this is the methodology, or the way you set up the reaction. So let us discuss mechanism how the selected region is amplified by using couple of cycles only, right? So I would like you to make your concept clear about denaturation, annealing and annealing step. What is the significance of these? How is the target selected by the primers, and what happens to the primer? What is the fate of primer? And you should also be able to identify type of amplification from this mechanism. So let us start with one double-stranded molecule, and let us see that we want to amplify this region shown here by the arrows. This is a pair of primer to amplify this region. So we will see how this region is selected and amplified. So what you have to do, reaction, and in each PCR reaction there are 3 steps. The reaction is also called as cycle. So let us see first reaction or first cycle. There are 3 steps. First is separation of a strand by denaturation at 94 degrees centigrade. So that will prepare the template. So denaturation prepare template as single-stranded DNA. However, the temperature is very high. It will not allow binding of the primer. Therefore, you have to cool down slowly. That is called as annealing. So the annealing ensures binding of primer. This is the step where you see hybridization. Once primers have bound, then Taq-DNA polymerase will extend the strand, the synthesis. So there are 2 things to note here. The amount has been doubled and become 2, and in the newly synthesized strand here and here, one end in each has been defined. We have to define both ends in both strands. Therefore, we have to repeat the reaction again. Cycle 2, same thing, 3 step, denaturation, and then annealing, and then extension. Here again, 2 things to note. Amount has been doubled, and in these 2 strands here, in the newly synthesized strand here and here, both ends have been defined, but we have to define the both ends in both strands. So you have to repeat the cycle again. So repeat the cycle again. Same thing, first step denaturation, then annealing, and then extension. Now again, couple of things to note. The first one is amount has been doubled here. It become 8. And if you see here, this is the product which is expected, which has been selected as double-stranded DNA. So you need minimum 3 cycles to see the appearance of the selected region. This region has been was selected, so this start appearing here, but the amount is very small. You cannot make use of this for any purpose. Therefore, what you have to do, you have to repeat the cycle again and again, maybe up to 20 cycles. Only then you will be able to see the product. So this type of increment or amplification is called as geometric progression or logarithmic progression. And this is because in each cycle, the product of each cycle become substrate for or the template for the next cycle. The other point is that the primers are shown by arrows, although they look like separated, but they are part of the newly synthesized strand. You cannot separate them. So what is the fate of primer? They become part of newly synthesized strand of DNA. So PCR amplification is geometric progression or logarithmic amplification, and this is simply because the product of each cycle acts as the template in the next cycle. And the amount of the product n formed from n, the initial amount n0 can be calculated using this formula. N is equal to n0 times 2 raised to the power x. X is the number of cycles. So n0 is the initial amount of template, n is the product form, 2 is the amplification factor, and x is the number of cycles. So using this formula, you can calculate and see that the template will be enormously or hugely amplified after 20, 30, or 40 cycles. So in summary, each PCR cycle has 3 steps. The first step is denaturation, which is normally conducted at 94 or 90 degrees centigrade. This temperature is not variable. This is almost fixed, so don't worry about this. Now this will allow the separation of the strands, but the temperature is very high. It will not allow binding of a small primer. Therefore, you have to cool down slowly. This step is called as annealing. Annealing is cooling down slowly to allow binding of the primer. And annealing temperature I have not shown here because this will depend on the type of primers, base composition, and the number of bases, right, in the primer. So primer will bind during annealing temperature, annealing step, and third step is extension, the enzymatic reaction, which is normally conducted at 74 degrees centigrade. This is very high temperature. This is optimal temperature for this enzyme. That is why it is called as heat resistant. It can survive. You can boil this enzyme for a couple of minutes and it will be active, and you will make the product will be doubled. So basically annealing temperature is very important, and it depends on the primer size and base composition. The question is how you will select or how annealing temperature is selected. So for that you have to have temperature. So how to calculate annealing temperature for a given primer? Annealing temperature is the temperature at which a primer binds to its target, and this temperature is selected based on the melting temperature of that particular primer. AT or annealing temperature must be at least 5 degrees less than the melting temperature or TM for a given primer. So this is very important to understand what is TM and how to calculate TM for a given primer. First thing, TM is the melting temperature at which 50% of double-stranded DNA is unwound. So basically, we should know how to calculate melting temperature or TM for a small DNA such as primer. So how to calculate TM of a given primer? First thing, you should know that the TM depends on the primer size and base composition. You can calculate for a small DNA or primer using this formula. TM is equal to 2 times A and T together plus 4 times number of C and G together in degree centigrade. So let me give you a practice and see if you can calculate TM for this given primer. So the primer is here given from 5 to 3 prime, and using the formula TM is equal to 2 times count the number of A and T together plus 4 times the number of C and G together, and put the value in this formula, and this comes out to be 50 degrees centigrade. So TM for this primer is 50 degrees centigrade. So annealing temperature should be less than Tm. How much less? 5 degree less. So annealing temperature will be Tm minus 5 degree centigrade. So this is how you can select the correct annealing temperature. If your annealing temperature is not selected properly, your PCR reaction is deemed to fail. So to amplify RNA, an extra step is introduced, which is called as reverse transcription. The product of reverse transcription is cDNA. And using this cDNA with a pair of primer, you can amplify any region by using PCR. So for amplification of RNA or RNA viruses such as COVID-19, you have to use a slightly modified PCR, which is called RT-PCR. So there are 4 applications I have listed. The first one is its application in understanding the disease pathogenesis. Second is in the development of diagnostic tools. And the third one is in the development of treatment strategies, including DNA, RNA vaccines or gene therapies, and also in DNA sequencing method. There are a large number of limitations or precautions, but one is very critical, and that is related to designing of the primers. As you know, the primers are designed based on the target sequence. Therefore, you must know the target sequence to design the primer and to use PCR. Without knowing the target sequence, PCR cannot be used. So thank you very much for watching the video, and I request you to like it, share it, and subscribe it. Until next time, take care. Bye bye.